name is Bagley. I'm a postdoc in the Forbes lab at the University of Iowa, and today I'm really excited to share with you some preliminary data that we've just gotten off that's just suggesting some really cool patterns. Okay, so the main goal of the Forbes lab is to describe why there's so much insect diversity in the world. And in particular, I'm really interested in why there's so much diversity of plant-feeding insects. So if you aren't aware, you probably are if you're here and you're an evolutionary biologist, but plant-feeding insects actually make up over a quarter of the described species, and that's probably an underestimate. And we think that this incredible diversity may actually be linked to the fact that they eat plants. So we think that shifts and subsequent adaptation to those new host plants may actually be a direct driver of insect speciation. However, there are some systems where the number of species of insects we observe actually exceed the amount of diversity we might expect based on the range of hosts that they use. One example of this are Bagley flies. So these are flies that will eat the sepal tissue of cucurbit plants. So cucurbits are things like squashes that you might be familiar with, although there's some more sort of exotic looking flowers down in South America where these guys live. And what we find is that these display the typical pattern of being very host specific. So this particular fly species will eat this species of flower, but not this species of flower. However, they take it to one more level. Instead of only specializing on one host plant species, they actually specialize on a particular flower sex of that particular host plant. So this species here won't use the female flowers, it will only use the male flowers. On top of this, however, you might expect, hey, okay, so each species of flower, each sex of flower has one fly species. No, we actually find multiple species of fly, all using the same flower sex of the same flower type. In fact, we sometimes rear multiple flies of different species from one flower, and the flowers are maybe like this big. So this is very unusual for what we might expect in plant-feeding insects. This pattern is really clear if you look at phylogeny, we map the host use onto it. So here I'm showing you a mitochondrial code 1 tree of different species of Bagley flies. And next to it, these bars here are colored to indicate what species of flower that particular species of fly uses. And next to it is a bar showing either what flower sex, so blue for male, pink for female, or host tissue is used. Some of these species also utilize the seeds of the flowers instead of the seed of the plant instead of the flowers. And just looking at it, you can see in most cases, most of these bars are entirely either one color or another color. Just to highlight this, here with species 1, 2, and 3, we see all three of them are using Gardenia acuminata, and they're all using male flowers for the most part. However, you've probably noticed that there are a couple of these that have a little bit more of a complicated host story, where we're seeing things using multiple species of flowers while still only using one sex, or things that are using a combination of different species of flowers and different flower sexes within both of those species. So today what I would like to share with you is some interesting results we found while we've been looking inside of each one of these species to figure out what's going on with these. Do they care if they're on different host plants? Is there any population structure between hosts? Or is there something else that's going on to drive the diversity in this group? Okay, so we took, like many others at this conference, a rad sequencing approach for this. But we didn't quite take a straightforward rad sequencing approach. We took a rad capture approach, and this was to get around the fact that some of our specimens are not quite in the best of shape, and so they can be difficult to recover the same markers from across different individuals. So using the Hoffberg protocol, we developed a pilot set of three rad data, which is just one of the many thousands of different iterations of dd-rad that exist in the world. And using 96 flies where we were able to get high-quality DNA out of them from anywhere from 2 to 10 different flies per species, we developed a set of markers that are our target markers. And from that set of target markers, we designed custom baits that targeted anywhere between 1 and 5,000 markers for each one of our fly species. We then captured those markers at a total of 635 flies. This is a map showing the distribution of all the different flies that we have, colored by what species. These are just species numbers here. And then that ranged anywhere from 41 flies to 130 flies per species, sort of spread across as much as we could of their range where we've collected them, as well as the host plant breadth that we had available at the time. And then we sequenced all of these on one HiSeq 4000 lane with 2x150 paraben sequencing. Okay, and then for each, I'm only going to show you a handful of species today. We just got this data back like last month, but we've already found some really cool patterns. For every one of these species, I went through and I called the markers out for each one, filtered them appropriately, and then put them into the program admixture, which is a population genomics population structuring method. In case if you've never seen the results of one of these things before, admixture will do two things at once. It simultaneously tests the number of populations in your data set and then assigns each individual to a proportion of ancestry for each one of those different populations you've inferred. So this is a typical admixture plot. Each one of these bars represents an individual, and the colors inside of each one of those bars indicate how strongly it's assigned to one of whatever number of populations you've tested. For Blepharonera species 10, our data best fit a model of 4 populations, so a K of 4. And you can see most of these individuals are assigning completely to one of those colors, one of those populations. This is really hard to interpret though, because you don't know who these individuals are or where they came from. So let's go ahead and put them on where they're from. So here's a map of South America, and what we can see is this block here actually corresponds entirely to individuals collected on this side of the continent, so in Bolivia, Peru, and Ecuador. This population corresponds to individuals on the other side of the continent, individuals collected in Brazil, French Guiana, and Suriname. And this white one corresponds to individuals collected in Trinidad and Venezuela. So what's going on here? We see it's a really strong geographic signal, and where might these have come from? So we actually have found this pattern before. A master student in the lab worked with Blepharonera species 10 with a pilot DD-RAD data set, and she found a very similar result with structuring, largely geographic structuring in her data set. She actually went in and she performed some demographic modeling using the program DIY ABC, and she dated the divergence of at least the Amazon and Guiana Shield populations to about the late Pleistocene. So unlike in North America where we had ice sheets that were advancing along, in South America you didn't have so much ice involvement outside of down here in like the mountains, but instead we had shifting of climates and shifting of current plant communities. So we took a look at what might have been going on in South America during the late Pleistocene, and we found this really exciting result. So number 1 here is areas where it was inferred that tropical rainforest, which is the habitat that Blepharonera occupies, and we see that there's really nice congruence here. We have this one really big one that could correspond to where we might have found Amazon flies hanging out at the time. We have another one here that might correspond to those Guiana flies that we found. And we also have a little one up here that might correspond to these central flies. So we still need to go back in with our new data and see if this is still the case with a better data set with more sampling, and if we include sort of explicitly these 3 populations, if we recover the same dates. But we're really excited to possibly find that some explanation for this really explicit pattern. So you may have also noticed I kind of left a couple of bars over here to the side, and this is because these aren't really popular, they aren't grouping with things the way we might expect. So most of these flies in these 3 groups here actually use the main host plant of the species, which is female Geranium spinulosa. But this population, while we have some individuals that use that main host plant, we also have a bunch that use the male flower of that host plant instead, that are forming their own population. But they're not all in the same geographic area, they're actually from both sides of the continent. So we're not quite sure what's going on with this. We think it could either be hybridization events or be driven by sort of a long distance dispersal that sort of allowed them to now expand their host range outwards and now be utilizing multiple plants, including shifting onto new ones, but it's really exciting. There's also this singleton individual here that actually was found on Geranium spinulosa seeds, and while it groups with ones from the Guiana side of the continent, it's actually from Peru. So this could be some more evidence of long distance migration events happening in this group. Okay. The other thing that's really cool is that we actually see very similar patterns in several other species that are also widely distributed throughout South America. So for Blepharonera species 4, we actually see exactly the same pattern where we see these 3 main populations that correspond to major geographic areas, which again might correspond to these putative Pleistocene refugia. And then we see this group of individuals that while some of them use their sort of normal host plant, male Geranium spinulosa, most of them are associated with unusual plants for this species, and again, are grouped on either side of the continent. We also see the same pattern, a similar pattern in Blepharonera species 8, where we recover our sort of Guiana population as well as our Amazon population. And in this case, we actually have 2 unusual host groups. So this one here, we have some individuals on the main host, 2 on other unusual hosts, splitting on both sides of the continent. And then this other group, some of them using the main host, some of them using an additional odd host, and again, splitting on both sides of the continent. And not to bore you with doing this a million times, but here again, we have the same pattern where we're recovering Amazon and Guiana group, and we also recover 2. This is actually a group made only of unusual hosts for this species, and we find that it does that same pattern of splitting and crossing both sides of the continent. And again, one that's the original sort of normal host for it, and then another weird one. So we're really excited for these. We're hoping to do a lot of demographic modeling moving forward, and we also have a couple of other species that I haven't included here today that don't quite have the same level of using a lot of different hosts, and we're really interested to see what kinds of patterns of population structure we might find there, if we'll only see sort of these geographic variations, or if we'll also see some like weird things where we're splitting across both sides on whatever unusual host instances we may encounter in those species. Okay, so with that, I would like to thank my lab who puts up with me doing all of this crazy stuff and coming in and showing all of my graphs all the time, being very excited. As well as the other members of the Dimensions project team. This is a really big NSF-funded project that does all sorts of different aspects of examining this really interesting blood-brain nerve system, including looking at their relationships with parasitoids and viruses that might be carried by those parasitoids, as well as phylogenetic questions about how these species are related to each other, as well as our own population genetics work. As well as people at my university that help me with sequencing and with doing both our high throughput sequencing as well as sort of co-1 sequencing, and our supercomputer folks for letting me do lots of hours of computations on their computer. And I'd also like to thank you, and I will take any questions you have. Yeah.
What are the characteristics that you're using to define the species?
So the species are defined using mitochondrial code 1 at the moment, and we do know that some of those are not, so we recently found that species 1 and 2 actually are the same species, and we're actually really excited about that for other reasons. That whole group up there we've thought of as being slow evolving. They seem to have been kind of doing the same thing for a long time, but otherwise we're using that.
It just seems like the geography or host might be useful in defining the species, and so it might look different.
Yeah, so I think that it's a valid point that we're still sort of figuring out some of the species designations. Pretty much everything we've done recovers the same species as we get with mitochondrial code 1. With everything, I've done a couple of dd-RAD-based trees with basically whole genome sequencing type data. We recover the same thing except for the species 1, 2 thing. But I think we're working towards being able to find morphological characteristics as well that we could compare to make it a robust species designation. Yeah.
That's a pretty nice job. So regarding your admixture plot, the group that had a really difficult geographic assignment, I wonder, so the admixture algorithm, because you constrain it to a k number,
yes,
it has to assign things to something, right? Mm-hmm. So I was wondering, for those that have weird biogeography, have you looked at more granular metrics? It could just be that assigning things to something, right?
So we're not just coming to a conference to present this. My typical thing that I do is I will repeat this with something like DAPC or some of the more spatially aware programs that do sort of simultaneous integration of looking for signals of isolation by distance on top of population structuring. So we're definitely going to be looking back and making sure, figuring out exactly what's going on with those weird sort of splitting populations. But this is what we're operating under for the moment. But if you have a program to suggest, I'm happy to use it. I'm always looking for new programs that can do a better job explicitly modeling